Hi students, welcome to my channel Atom Bio 360. So students, in today's class I will discuss about glycolysis pathway. So in last class we discussed about types of the respiration based on the substrate and based on the availability of oxygen. So in today's class we will discuss about glycolysis pathway. So basic information about glycolysis also we discussed in the last class. So now I will start this glycolysis pathway. See here, glucose. So how many carbons is present in glucose? So the glucose contains 6 carbons. So 6 carbons is present in the glucose. So the glucose is converted into the glucose 6 phosphate with the help of the enzyme hexokinase. So in this process, 1 ATP is consumed in this reaction. So ATP means how many phosphates? 3 phosphates. It is converted into ADP. So 1 phosphate is transferred into the glucose. So glucose is converted into the glucose 6 phosphate. Why we will say this is glucose 6 phosphate? Because the phosphate is attached to the 6th carbon atom of glucose. So because of that we will say this is glucose 6 phosphate. So glucose is converted into the glucose 6 phosphate. Again, the glucose 6 phosphate, it is converted into the fructose 6 phosphate. Okay, so glucose and fructose. So both are isomers. Okay, isomers means same molecular formula and different spatial arrangement is present. So that kind of the molecules is called isomers. So glucose and fructose both are called isomers because both having a same molecular formula but both have different spatial arrangement. So because of that we will say the glucose and fructose both are isomers. So this is in this step the enzyme involved in this step that is called isomerase. So this is the second enzyme in the glycolysis pathway. Okay, so then the glucose 6 phosphate is converted into fructose 6 phosphate. Again, fructose 6 phosphate is converted into fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. This is not diphosphate. So what is the difference between bisphosphate and diphosphate? Suppose here see ADP. What is the abbreviation of ADP? Adenosine diphosphate. So why it is called diphosphate? Because 2 phosphates is attached to each other. So if the 2 phosphates is present in the successive atoms, then that is called diphosphate. But here 1 phosphate is present in the first carbon and 1 phosphate is present in the 6th carbon. So because of that this structure is called fructose 1,6 bisphosphate. So here also in this reaction also 1 ATP consumed. So see here ATP, it is converted into ADP. Okay, so 1 phosphate transferred from fructose 6, that is ATP to the fructose 6 phosphate. Then 1 phosphate transferred from ATP to the first carbon atom of the fructose. So because of that the fructose 6 phosphate is converted into fructose 1,6 bisphosphate. So here another enzyme is involved in this step, that is called phosphofructokinase. Phosphofructokinase. So here see these 3 reactions. So 3 enzymes involved in these reactions. So in these 3 enzymes the first enzyme is there. So the first enzyme is called a regulatory enzyme. Okay, so the glycolysis is regulated by the hexokinase. Why the glycolysis need to regulate? So that is, suppose excessive glycolysis is present in the body, it will produces more amount of energy. So unnecessarily more amount of energy is produced, that is not good for the body. So that is not required by the body also. So the glycolysis speed, it is regulated by the enzyme, that is regulatory enzymes. So hexokinase is the first regulatory enzyme, and another regulatory enzyme is present. So that is the second regulatory enzyme in the glycolysis, that is phosphofructokinase. This is the second enzyme. So kinases means this is a class of enzymes belongs to the transferases. So it will transfer phosphate from ATP to the glucose. So because of that, this is called kinase. So here also this is kinase. Here this enzyme also transferring phosphate from ATP to the fructose 6-phosphate. So because of that, the kinases is called transferases. That is class of enzymes, one class of the enzymes. So fructose 1,6-bisphosphate is produced. So again, in this reaction, one ATP molecule is also consumed. So two ATP molecules consumed in the first reaction and the third reaction. So because of that, this phase is called energy activation. So that is here see. So this phase is called activation phase. So why, students, I am asking one question. So in this place, generally what is the target of glycolysis? Glycolysis for production of energy or consumption of energy? What is the ultimate goal of the glycolysis? Ultimate goal of the glycolysis, that is production of energy, not consumption of energy. But see this step and this step. So in these two steps, energy is consumed or energy is produced? Yes, energy is consumed in this reaction and in this reaction also. So that is ATP consumed. Here also ATP consumed in this reaction. Okay, so why ATP is consumed in this reaction, in these two reactions? Because glucose is chemically inactive molecule. So the inactive glucose, it is activated with the help of ATP. So because of that, this phase is called energy activation phase, or this is called activation phase. So chemically inactive glucose is converted into the active molecule with the help of this ATP. So because of this, two ATPs is required for activation of glucose. Okay, so after activation, the fructose 6-phosphate, that is fructose 1,6-bisphosphate is there. So it is converted into the glyceraldehyde, two molecules of glyceraldehyde 3-phosphate. Two molecules of glyceraldehyde 3-phosphate produced from the fructose 1,6-bisphosphate. So how many carbons is present in fructose 1,6-bisphosphate? See here, 6 carbons is present in the fructose 1,6-bisphosphate. So fructose 1,6-bisphosphate break down into the two molecules of glyceraldehyde 3-phosphate. Two molecules, 2 into glyceraldehyde 3-phosphate means two molecules of glyceraldehyde 3-phosphate. So the two molecules of glyceraldehyde 3-phosphate, one molecule of glyceraldehyde 3-phosphate contains 3 carbons. So 2 into 3, totally 6 carbons. Okay, so one enzyme is involved in this reaction. That enzyme is called aldolase enzyme. Okay, aldolase enzyme is required for this step. So fructose 1,6-bisphosphate is converted into the two molecules of glyceraldehyde 3-phosphate with the help of the enzyme aldolase enzyme. So this enzyme is called pacemaker enzyme. So pacemaker means that is it is important for breakdown. Okay, so breakdown of fructose. So fructose is breakdown into the two molecules of glyceraldehyde 3-phosphate. So again this glyceraldehyde 3-phosphate is there. So this glyceraldehyde 3-phosphate again it is converted into the dihydroxy acetone phosphate. Dihydroxy acetone phosphate. Okay, so glyceraldehyde 3-phosphate again it is converted into two molecules of dihydroxy acetone phosphate. So simply you can write DHAP. dihydroxyhistone phosphate, 2 molecules. So both are isomers. So glyceraldehyde 3-phosphate and dihydroxyhistone phosphate, both are isomers. Okay, so which enzyme is important for this reaction? Yes, your guess is correct. That is isomerase enzyme. Glyceraldehyde 3-phosphate isomerase. Okay, so glyceraldehyde 3-phosphate isomerase enzyme. That enzyme is important for conversion of glyceraldehyde 3-phosphate into dihydroxyacetone phosphate. Okay, so again the dihydroxyacetone phosphate also chance to produce chance to convert it into the glyceraldehyde 3-phosphate. Okay, so this is the energy activation phase. So students, in next class we will discuss about energy extraction phase. That phase is responsible for the production of ATP and NADH2. So this phase is important for consumption of energy. Why energy is consumed? That inactive glucose molecule is converted into the active glucose molecule. So in this I explained about 2 regulatory enzymes. So hexokinase is one regulatory enzyme. So it will control the rate of glycolysis, and another one is phosphofructokinase. So this enzyme is also controls the rate of glycolysis. So phosphofructokinase is the principal enzyme for regulation of the glycolysis. So glycolysis is mostly regulated by the phosphofructokinase. So because of this enzyme is called principal regulatory enzyme in the glycolysis. So in next class I will explain about energy extraction phase. So students, I will meet in next video. If you have any doubts, comment your doubts in the comment section. If you like the video, please click the like button and subscribe my channel and share these videos to your friends also. Okay, thank you very much for watching my video.